Alright, the first thing we're going to go over is a remarkably simple concept, which I think everybody who's actually paying attention will understand. It's called selective breeding. And what that is you select parents in order to get desired or certain traits in the offspring or the babies. So, for example, there are hundreds of different breeds of dogs. All dogs came from the gray wolf. No matter what kind of dog you have, whether it's a poodle, a German shepherd, doesn't matter, they all originated as a gray wolf. But they've been selectively bred over hundreds and hundreds, and if not thousands of years, for those dogs to get different traits. Some of them have real long hair, some of them have very short hair. Whatever traits you want in your dog, that is what you breed into them by selecting certain parents. Chickens are also selectively bred in order to get hens that produce a lot of eggs. If you're selling eggs at a supermarket, you want your hens, the female chickens, to produce as many eggs as possible. Well, chickens have been selectively bred over hundreds of years, so now the hens produce far more eggs than they used to produce in the wild. Another thing, plants are also selectively bred. You can see up here, these are wild tomato plants. Not very big, it's almost like the size of like a cherry. But over the years, tomatoes have been selectively bred, so their fruit is much, much larger. Here is a wild carrot plant. You can see that little tiny thing you'd have to eat if you ate a wild carrot. But over the generations, carrots have been selectively bred, so the part of the carrot that you eat is much, much larger now. Here is a wild banana and its fruit. And here is the more familiar banana that you'd buy in the supermarket that has been selectively bred, so the fruit is much larger. And that is what selective breeding is. And now there's two different types of selective breeding. The first one we're going to go over is called hybridization, otherwise called outbreeding. And this is when you're crossing two parents that are very different from each other. So these parents have very different traits, and what you want in the offspring or the babies, you want them to have the best traits from both parents. An example of this is if you grew strawberry plants. Strawberries, their fruit of course tastes really, really good. But when you raise or grow strawberries, if the temperature gets too low, the strawberry plants will die. So what you could try to cross-pollinate your strawberry plants with a different plant that, say, lives maybe in Alaska that can survive in the snow. Now you're trying to get the good things from both parent organisms, so you want the good tasting strawberries from your strawberry plant, but you want the trait of being able to live in cold temperatures in the snow in the offspring. So you want both, good tasting strawberries and being able to survive cold temperatures. That would be an example of hybridization or outbreeding. Now, what are the advantages? Number one, if you're lucky, you can combine the desirable qualities from two different parents. So in the example I just mentioned, you might be able to get plants that produce very good tasting strawberries, and they're able to survive cold temperatures. Number two, these organisms, these plants, these animals are usually healthier and larger. So for people that have dogs, if you have a purebred bulldog, let's say, those animals are usually less healthy than a mutt that you could buy at the pound. The reason is, when you inbreed, this is what we're going to go into next, those organisms are less healthy, but when you hybrid or outbreed, they're usually larger and healthier. And in wild populations, when you outbreed, you're increasing genetic diversity. So you're making all the individuals in the population a little bit more different, and that is important for those populations to survive and adapt over time, and that's what we're going to go on in when we get into evolution later this week. And now some disadvantages of hybridization or outbreeding. It is more difficult to do. If you're only concentrating on one trait, say like having cats with very, very short hair, that is kind of easy to do. You find a male cat and a female cat that have short hair, and chances are their kittens are going to have short hair. But when you're trying to outbreed or hybridize, it's more difficult to get 2 or 3 desired traits from 2 different types of parents. And number 2, if you cross members of different species, their offspring are often sterile, which means they cannot reproduce or have offspring of their own. An example of that is the mule. So if you want a little baby mule, you don't get 2 mules to have a baby. No, you get a female horse and you crossbreed it with a male donkey. And then that's how you'll get a mule, the hybrid. Also, it was an accident, but you can get a liger, which is made from a female tiger crossbred with a male lion. Now, they didn't do this on purpose. The male lion actually jumped the fence and got in with the female tiger, and it produced a very, very large kind of half tiger, half lion called the liger. Okay, and now the other type of selective breeding, which is not outbreeding, but inbreeding. That's when you're crossing 2 similar organisms with very similar genotypes, which means they have very similar genes. These organisms are often related. So we're not talking about people here. We're talking about farm animals, plants, things like that. So the advantage of this is, first of all, it's much easier to do. It's much easier to get the desired traits you want when you're crossing 2 organisms that are very similar. An example of this is if you cross 2 purebred bulldogs, all the little puppies, they're all going to look like bulldogs. I mean, there may be tiny little variation. Some might be a little bigger, some might be a little smaller, but their colors are all going to be pretty much the same, and it's going to be kind of hard to tell one from the other. On the other hand, if you cross like 2 alley cats, like 2 stray cats, and they have 10 kittens, some might be orange, some might be black, some might be gray and white, who knows? You get a bunch of different things. But that would be outbreeding. But with inbreeding, you pretty much know what you're going to get. Another advantage is that the traits can actually be accentuated or enhanced over time. For example, like for dogs, poodles are a pretty big dog. Let's pretend like that. I don't want to draw a dog. Let's pretend like that circle's a dog, a poodle. Well, if you keep breeding the smallest puppies you got, they can keep getting smaller and smaller. Next thing you know, you have what's called a toy poodle, which is a pretty small poodle. But they kept even one of them smaller. They kept breeding smaller and smaller, and then you end up with what's called a teacup poodle. So with inbreeding, you can accentuate or enhance the trait you're looking for to get smaller and smaller and smaller, or bigger and bigger and bigger, or faster and faster and faster, whatever you're looking for. If you're looking for one trait, it's easier to enhance that trait. Disadvantages: harmful traits from the recessive alleles. Frequent inbreeding makes the offspring less healthy. They're more likely to have disease. And the opposite of outbreeding, genetic diversity or variability is reduced. So in wild populations, they have a decreased ability to adapt over time if there's some sort of environmental change. And that'll be in evolution. And just a picture that shows you how all the dog breeds have come about over, thousands of years. All dogs on Earth started as that, a gray wolf. And through selective breeding, they have turned into all these different breeds of dogs that do not even really resemble. I mean, this little guy came from that vicious wolf. I mean, that's crazy. But through selective breeding, the wolf turned into that. The little dachshund with his tiny little short legs and this fancy poodle. So selective breeding turned the wolf into all the breeds of dogs. And it doesn't really take too long. In 1790, which was just after the American Revolution, that is what the English bulldog looked like. And today it looks like that. So it looked pretty athletic back there in the late 1700s. And today, this very cute dog, but not very athletic. He's got the little short legs. He's very wrinkly. He gets tired if he runs like 10 feet. And these guys are not healthy at all due to inbreeding over several generations. And now on to real biotechnology. For thousands of years, humans have been trying to manipulate the DNA of plants and animals through selective breeding, and they still do it today because selective breeding is very cheap, but we do have the ability to now use genetic engineering. So we can actually skip the selective breeding part and go in to the organism's DNA and tinker with it. You can identify, change, remove, or add genes at the molecular level. So you can skip the selective breeding part. You can just go right into their DNA and get exactly what you want. So one thing you got to know, restriction enzymes. Remember, those are like the little scissors. They're made out of proteins, they're enzymes, and they cut DNA at precise locations. There are over 600 different types of restriction enzymes. They're naturally found in bacteria. You won't need to know that. But just know the restriction enzymes cut DNA at precise locations. And hopefully you kind of remember or have heard of this, gel electrophoresis. That is a technique that is used to separate the fragments of DNA once they have been cut by restriction enzymes. A little more on restriction enzymes. This is the one called EcoR1, which is like one of the most famous ones. And EcoR1 cuts DNA at an exact spot. It's looking for this specific sequence of GAATTC. Every time, and this little guy right here, that's the restriction enzyme, every time it sees that exact sequence of bases in DNA, it cuts between the G and the A, and then cuts again between the G and an A. Now remember, whenever it leaves this little single strand at the end, that's called a sticky end. Sticky end, the TTA is going to go perfectly with the AAT. So those things can go right together, and then you have, once they come together, this foreign DNA, which has been spliced on to the natural DNA, you have what's called recombinant DNA. So recombinant DNA is DNA that has had genes inserted into it artificially in the lab. All right, we already talked about this, but let's talk about it one more time. So what is recombinant DNA? Recombinant DNA is artificial DNA that is created by combining DNA sequences that would not normally occur together by splicing genes together in the lab. So when we did that little with the scissors and the tape where we cut out the human gene for making human growth hormone and then we inserted it into the bacterial plasmid DNA, that was an example of how to make recombinant DNA because the bacterial DNA now contained the human gene for making human growth hormone. Therefore, it is now called recombinant DNA. All right, now the last slide. Transgenic organisms. Transgenic organisms. So this goes with the genetic engineering that we saw on the last slide. These are organisms that contain DNA from another species which have been incorporated into their DNA through genetic engineering. So this is where a gene is actually cut out using restriction enzymes. They cut the gene out from some other organism and they insert it into a fertilized egg to change the DNA of that host organism. For example, they can get plants that glow in the dark. Well, how did they do that? Well, they took out the jellyfish's gene for glowing in the dark and they inserted it into a plant and voila, that plant can now glow in the dark. Here's another example. Here is a regular looking orange on the outside, but that orange had the DNA of kiwi, a separate type of fruit. It had kiwi DNA inserted into its DNA, and now on the outside it looks like an orange, but that orange produces fruit like a kiwi because it had a gene for kiwi fruit inserted into the orange DNA. And this is where sometimes on the news or in the newspaper or wherever you'll see concerns about GMOs. genetically modified organisms. And some people are afraid that by changing the DNA of fruits and vegetables, things we buy at the store, that those may become harmful to eat. So that's what genetically modified organisms are.